Hello, everyone, and welcome to today's webinar, How to Advance Cell and Gene Therapies with an Innovative, Flexible Approach. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Dassault Systemes. To learn more, visit 3ds.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. All questions will be answered via email following the broadcast today. You may also submit any technical issues if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the Continuing Education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speakers, Chris Andrews, Senior Solution Consultant at Dassault Systemes, and Domenico Palumberi, Solution Consultant at Dassault Systemes. Chris and Domenico, you may now begin your presentation.
Good day, everyone. My name is Christopher Andrews. I am a solution consultant with Dassault Systemes, and joining me today is my colleague,
Domenico Palumberi. Hi, everyone. I'm solution consultant for Dassault Systemes and covering life science.
Thanks, Domenico. We're going to be talking to you today about advancing cell and gene therapies from the beginning of the drug discovery phase all the way through commercial manufacturing, and how you can improve those various stages of that manufacturing development process through innovative, robust, flexible solutions. So we're going to start with Domenico.
Yes, thanks. So the cell and gene therapy market is having a boom recently. I mean, you can see here the number of cell and gene therapies launched globally by the year. I mean, it's growing a lot. And as well, like the sales growth trends is growing a lot. And there are like really nice prediction from now to the 2024.
While the sales trajectory is skyrocketing, we're also seeing that the cost of manufacturing these products is also going up, and the types of companies that are getting into cell and gene therapy aren't always the pharma giants. They're oftentimes much smaller companies, molecule shops, as they're sometimes called, and having the resources to be able to make those products efficiently, cost effectively is going up as well, and it's very difficult to do.
Yes, indeed, and this is completely related with the difficulty of the process itself. As we can see in the next slide, I mean, the manufacturing process is first of all a huge collaborative test, in which you need to collaborate between academia, R&D institutional, hospitals, CDMO, pharma, and as well patients. So, I mean, looking at this scenario already, I mean, we can see that collaboration is a key component of this manufacturing. We go and we see the step of the different of the manufacturing cell and gene therapy. It's very complicated because you start with apheresis that can be from patient or donor. You froze down the cells, you put an airplane, and you send to the pharma, where start the cell modification, right? Usually with a viral vector that are arriving maybe from your collaborator, from a CDMO and CMO. So you genetically modify the cells, and after the step, you start the expansion and the harvest of these cells. When you finish all of these, you have all the quality assurance and testing that you need to do on these cells. At the end, you froze down back and you send to the hospital when there will be the infusion to the final patient. So, I mean, it's a very long process. It's a very complicated process. Think about the fast one is around 24 days, just to give some numbers from the beginning to the end. So, I mean, this requires a lot of effort in sense of a lot of scientists working on it, working in controlled environment as well, for example, clean room or isolators. So working very tight between them with a huge responsibility on the shoulder, because at the end, if something gets wrong, I mean, the patient will not receive the therapy itself.
Indeed. And this is a highly specialized process. This is different than any other type of drug product that is made at scale in the industry. So there's been some learning that's been happening at the same time as the development of these products has gone on, right? If you look at the typical stages of a therapeutic manufacturing, or from conception all the way to essentially the patient arm, you've got these major phases. You've got the basic research that happens, drug discovery. It will go to preclinical for dosage studies and for tolerability studies. And then it'll go to human trials, and then it will eventually go to commercial manufacturing. A lot of that happens in a much more compressed time frame. With cell and gene therapies, basic research and drug discovery can be more focused with cell and gene therapies because you've got a target indication that you're looking for. Maybe it's a gene sequence that you know that you need to alter in order to deliver the therapeutic to the patient. However, that is an extremely costly and time-consuming process, even when you do know what your therapeutic target is with that genotype. So having a software solution that can reduce the complexity of that challenge, finding candidate processes or molecules or whatever that would be amenable to that therapy, can be greatly reduced. If you've got a target performance profile or certain attributes, whether it's the end viscosity of that infusion, or a binding affinity for a particular molecular target, you can use software, you can use a solution to reduce that complexity and then bring you further into those later stages. Being able to go from preclinical onto clinical, being able to scale up those batches from just the test article to something that's larger scale for a larger patient population. That's going to require a lot of documentation. It's going to require a lot of data. Particularly once it gets into the human scale or the human scope, having data traceability and document traceability, and also making sure that all procedures have been followed all along the way. That's absolutely critical for getting things past those regulatory hurdles, but also ensuring patient safety.
And Chris, do you think that the paper can be an efficient method in order to follow all these procedures?
Unfortunately, I'd have to say it is not an efficient way of doing these things, but it is extremely common even in the very large pharma companies. You're still seeing paper, batch records being captured, paper being exchanged even within groups within the same company. And often these smaller cell and gene therapy type companies are relying heavily on their partners, contract manufacturing organizations, contract research organizations. And though those entities may individually be using digital systems, they're not talking to each other. So you have a lot of siloed data. So having something that will enable collaboration would really make the difference, especially for the patients that are relying on these therapeutics.
Yeah, let's think about the compliance. I mean, look at this. The recent FDA compliance issue fall into the majority fall into these 5 categories, right? And all of these 5 categories are related to the usage of paper. So think about how, usage of digital solution can help to be compliant with regulation.
Yes, indeed. So you've got the compliance challenge in terms of following procedures, documenting that data traceability. And in GMP manufacturing as well, there are certain challenges that come along with that. creating those protocols, creating those SOPs that go along with your process, being able to maintain them to make sure that they're current and that they're matching the actual process that is in use. Being able to store these records that you're generating, whether it's your quality system, your learning management system for the procedures, but also the data that's being generated as part of the manufacturing process. Sample tracking and chain of custody as it goes through. All of that is absolutely critical in the cell and gene therapeutic space because, oftentimes if it's autologous cell transplants, you have to have, the identity that the cells are going right back to that donor. Again, we talked about siloed data. It's making it very difficult for people to work efficiently, to have that data traceability.
Yeah, because let me say one thing about this. I mean, we're talking about paper, right? But some of the companies start digitalization, of course. I mean, but they have different pieces of software all around. These different pieces of software cannot talk between them. So the data that are gained from a software cannot be transferred to another solution. This results in data that are not usable anymore, and this means a lot of richness that is going away. Because data analysis is like, a richness of a company.
Indeed.
So
if they don't have the whole story, they're not able to make their full submission or get the most value out of the information that they've actually generated.
So, but how digital solution can help, like cell and gene therapy manufacturing, especially in GXP environment? The first thing is helping with writing procedures, so helping writing down the SOPs. SOPs must be written down in a universal language. So, for example, following S88 standard. So, in order to improve collaboratives, so you can send out to your you can send out to your collaborators like the protocols, and they can read because it's in the same format. After that, you need to be sure that what you are writing down in the SOP is executed in a good way by scientists. Scientists are working under a lot of pressure. They know that if they wrong, the therapy will not be delivered to the patient. They are working in a clean room or using isolators, so in very tight conditions. So they have a huge pressure on them. So everything that can facilitate the execution of the SOP is important. And how can be facilitated the execution? First of all, in a step-by-step execution in which the scientist can just follow what to do. Automatic and embedded calculation, for example, so that will allow the scientist do not stop and do calculation, but just follow the procedure. And instruments that are directly interconnected with the software itself. So, for example, if I'm running my fax analysis, the results of the number of positive cells will arrive directly to my software, and from there I can proceed to the next step. And this is not important just for execution, so to reduce the human error, of course, but it's important as well for the compliance, because as we know, all the documentation is very important for compliance reasons, right? Every step must be recorded, audit trail must be created, and all the raw data must be saved for every step of this. And when you work in GXP condition, it's not just this. You need to take care as well of all the metrology, all the logbooks, and already avoiding all of that job to the operators, to the scientists, is a great advantage. Another important point is that all of the solution must be easy to be deployed. So there must be an easy implementation of the solution, because scientists must work on science, should not work on IT solution, implementing IT solution, right?
Yeah, definitely. You want the solution to fit your process. You don't want to have that learning curve be so high that ultimately the users aren't going to use the solution, and that will open you up for a non-compliance. And, it's a risk to data integrity. The other thing also, you want to be able to collaborate doing analytics, right? you need to have access to that data. Not only is digitalization of your procedures, your processes, your data collection, not only does that minimize human error and ensure that you've got that auditability or that provenance of the data, but it'll also give you access to that preponderance of data that you've generated, making your therapeutic. And what that will do is not only allow you to do things like continued process verification to make sure that it is going according to your manufacturing process or the acceptance criteria for that particular product, but it'll also enable you to do process improvement, where you can say, hey, this particular batch was running a little closer to our specifications than normal. Let's dig into that. Let's find out why that is. So you can start pulling in that data, not just internally, but with those collaborators as well, those folks that are in your contract manufacturing network or your contract R&D areas there. It's a huge advantage for these organizations.
It is indeed. Summarizing a bit what we have been discussing with, which are the key advantages in using IT solution in cell and gene therapy. First of all, due to the nature of the manufacturing, facilitating the collaboration between the different departments, different laboratories, different entities, it's the key. A good collaboration means improve in quality. As well, data integrity is another big advantage. Of course, there is a lot of regulatory on cell and gene therapy, since, we are talking about injectable in the patients. And being able to automatically save all the results, automatically save all the raw data is a really, really a great advantage for this reason. And it's removing work from scientists. So the big advantage is that scientists can be much more dedicated to them patient, to them, the work that is indeed to do science.
That's right. I've heard from a lot of former peers in industry, but also once, now in my role at Biovia and Dassault Systèmes, I go to prospective customers or existing customers, and I'm like, I didn't do a Ph.D. so I could copy and paste into Excel. And I'm like, I hear you. So yeah, it's absolutely bring up those very skilled professionals to be able to focus on the very important work and not do non-value-added work. Lastly, I think one of our takeaways from this whole talk, it's about having a holistic view of that process. So from the very beginning of when you start narrowing down those candidates for your therapeutic, all the way through the preclinical, clinical, the hospital setting or the caregiver setting, getting the data from the patient, or getting the cells and data from the patient and bringing it completely through the process, being able to understand everything so that you can make it all better, more efficient, more cost effective. That is one of the key reasons we do what we do. And it really can be a game changer.
So, and which are the message that we should take away, which are the key takeaway from this discussion? First of all, that IT solution can enhance and facilitate the collaboration efficiency. Enhancing efficiency means as well reducing the cost. This is a really important point. Cell and gene therapy manufacturing has a very high cost at the moment. So being able to facilitate this workflow means reducing, of course, the cost. And a lot of costs are as well dedicated to data integrity. Think about saving all of the raw data, saving all the log files, all the important data for compliance is taking a lot of effort in time and money, of course. So being able to do this in automatic manner is a huge advantage for the cell and gene therapy manufacturing, definitely.
And certainly bringing a digital paradigm to all of this work that happens from the early discovery phases through to collecting cells from the patient, bringing it to the clinical space, or when it has to be scaled up and brought back to the patient, having a holistic view of all of the data that's generated throughout that process. It affords not only continuous improvement so that you can understand, hey, this particular batch is we're running closer to our design criteria, closer to our control limits than we wanted. Let's investigate and see how we can make this better. So you've got that continuous improvement element. But what that also allows you to do when you have access to all of that data, it allows flexibility, which is absolutely critical for a cell and gene therapeutic, because either that condition or that patient population, there's a lot of variability in there. You need to be able to adjust as you go. Having all of those be digital, it's more efficient, sometimes not even possible if you're just using paper or siloed data systems. So having everything integrated allows for that flexibility. And again, that flexibility affords efficiency and savings. And with that concludes the presentation portion of the session. So I think we're ready to move on to our Q&A. Thanks so much for your attention. Looking forward to your questions.
Thank you.
Thank you, Chris and Domenico, for your informative presentation. We would also like to thank LabRoots and our sponsor, Dassault Systèmes, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. As a reminder, any questions submitted today and during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.